good.
Maybe we'll save some time. Because
Maybe
we're
we'll
like,
get it.
hello, week 4 is over. Yay, it's Thursday. I'm so happy. I love weekends. Oh my God. My life is great. Especially on weekends. Anyway.
Yeah. We love weekends too.
When you have a little more money, it's really good. But, so, we only have week 5 left and 6 left, and then your final exam. So, we can't really doubt it. All right, so, we're going to talk about genetics. It's the genetics chapter. But it's really not about genetics. It's about anabolism. Because remember, we're talking about metabolism. And I'm talking about microbial, bacterial metabolism, prokaryotic metabolism. It's a little bit different from eukaryotic metabolism, which you might have had in A&P, or you might not ever have had. But at any rate, we're talking about anabolism. There's 2 kinds of anabolic reactions we're going to look at. One is DNA replication, and the other is protein synthesis. I had written on the board a couple of days ago, DNA replication occurs when the cell is getting ready to divide, because it has to have a copy of the DNA for each new cell. Protein synthesis happens when the cell is making proteins. And remember, bacteria make proteins for the cell's own use. So they're not going to copy the entire DNA to make a protein. They're just going to copy the gene that codes for the protein that they need. So, we'll come back to that later, and right now we'll focus on DNA replication. The model of DNA replication that scientists have settled on is called semi-conservative replication, and you'll see why. All right.
Is it the plasmid?
No. I'll explain it. Here's your bacterial cell. Plasma membrane, cell wall, it's got some ribosomes in there, it's got a nucleoid. Some bacteria have a plasmid. The ribosomes we don't need right now for DNA replication, but they would be in there, just wanted to remind you, and when we do protein synthesis, we'll need the ribosomes. Right now, we're going to focus on the genome of the cell. The genome is all the genes of the cell. It's the DNA of the cell. It's all the genes in its nucleoid, and if it has a plasmid, it also counts as part of the genome. When a bacterial cell replicates its DNA to replicate itself, it will replicate the plasmid as well. We're not going to worry about the plasmid right now. Just know that the plasmid can also be replicated. In genetic engineering, what they actually do is they get the cell to replicate multiple plasmids, and then they can put the plasmids in other cells, but that's a whole other thing. So for DNA replication, to explain it, we're just going to focus on the nucleoid, okay? Just didn't want you to forget about the plasmid. So right now, we're not going to think about the plasmid. We're just going to focus on the nucleoid. So this is the nucleoid. This is my picture of the nucleoid. So when you look at a bacterial cell, all you see is a dark staining area, perhaps, if you can see inside the cell. And it's a closed loop of DNA. So I've taken it out of the cell and made a simplified drawing of it. Okay? It's a closed loop of double-stranded DNA. And it has little hydrogen bonds holding these loops together. Whenever I write a hydrogen bond, I write a little dotted line. Because hydrogen bonds are weak attractive forces, easily formed and easily broken. The purpose of hydrogen bonds in biological systems is to give the molecule resiliency. And you know, DNA is a molecule, right? It's an organic molecule, it's a nucleic acid. It's held together with hydrogen bonds, because hydrogen bonds allow the molecule flexibility so that it can be copied. We'll get into that a little bit more in a minute. Okay. If I'm going to explain DNA replication to you, I either have to be a much better artist or I have to go to the PowerPoints. I don't want to do either one of those things right now, so you're going to have to live with my primitive drawings. So to simplify things for myself, I cut the loop of DNA open and laid it out, okay? But just remember, DNA replication would occur while it's in this form. It would go around the loop. If I forget, remind me, I'll make you a little drawing of that. Right now, we're gonna look at the structure of the DNA, which I wish I didn't have to go into all of this because this is something I think that should be covered in the chemistry primer, but anyway. So, it's still too hard for me to explain it in this twisted up structure. But why do we call it the double helix? Because it's helical, and there's two strands of nucleotides twisted together. So it's a double helix. Who coined the term double helix?
Two people, I forgot. There was two of them that were working.
Forgot their names. And who did they steal the credit from? Thank you. A woman. No offense, fellas. Actually, Rosalind Franklin had it figured out what the sides were made up of. She didn't understand what was going on in the middle. That's the part that they were able to put together that she couldn't figure out. So I'm going to show you what that is. So I'm going to have to enlarge it. So bear with me. I'm going to write something a little bit hairy up here. I'm going to write sippy. 1 sippy, 2 sippy, 3 sippy, 4 sippy, 5 sippy, 6 sippy, 7 sippy. Think I can fit 9 sippy? 8 sippy. 9 sippy. I can't fit 9. 1, 2, 3, 4, 5, 6, 7, 8. I wanted to squeeze 9 in there, but I guess I didn't do it. We'll do 6. 6 will have to be enough.
Should there be 9?
Well, you know what? Whatever it is. All right. Now. Oh, got questions. All right, my artwork's not so good today. I might end up erasing off the bottom one, but whatever. Oh, wait, I made a boo-boo. These are NBs. Doesn't feel right. Right, let's pretend I got another one down here. S-I-F-I-S. All right, Alright, let's pretend I got another one down there. I didn't really get it good, but it's close enough. Alright. Now remember, this is actually in this form, which is a simplified form of that, but we've stretched everything and flattened everything so you can see it blown up. Now, I can't fit the entire DNA on the board. So this thing is technically going much longer. I'm just showing you a portion of it. All right, so this is your DNA molecule. So what do I have right here? Good, a nucleotide. And then here's another one. Here's another one, here's another one, here's another one, here's another one. So I have 2 strands of nucleotides twisted together helically. They're both twisted together. That's why I got a double helix. And they're held together in the center by hydrogen bonds. So these dotted line bonds, those are hydrogen bonds. Easily formed, easily broken, gives the molecule resiliency. If I spelled that correctly. These bonds here, the SIPI bonds, and this bond here, those are covalent bonds. The covalent bonds are strong bonds sharing of electrons. These bonds will stay together when the hydrogen bonds break.